This video is chapter 24, the immune system notes. An immune system response is a blood-based response. So organisms that don't have blood are not immune to things. Back in first semester, we learned about antibiotic resistance and how bacteria can be resistant to an antibiotic. We would never say that bacteria are immune to an antibiotic. Bacteria, of course, are single-celled organisms that do not have blood, and thus immune and resistance is not the same thing. Resistance just means that the chemical has no impact on the cell, whereas immunity means that there is a blood-based response against a specific pathogen. So we'll go through that here now. When we talk about the immune system, we are talking about a blood response. That blood response has to do with white blood cells. Your blood is made of four main components, one of which is red blood cells. Red blood cells deliver oxygen. The purpose of that oxygen is to make ATP. Oxygen is the final electron acceptor in the electron transport chain inside the mitochondria to make ATP. Good throwback to first semester. So when we talk about the immune system, we're not talking about red blood cells. We are talking about white blood cells, those that fight pathogens. A pathogen is anything foreign that is inside of your body that doesn't belong. Platelets, you may have heard of. Platelets are used in clotting your blood so you don't bleed to death from something like a small injury. And then the fourth component of blood is plasma. Plasma is a liquid that maintains proper salt and water balance in all of the blood cells, and also serves as a liquid through which the blood cells can move by the action of your pumping heart. So here, chapter 24, our focus is white blood cells. White blood cells also go by the name leukocytes. You may have heard of leukemia. Leukemia is a cancer of the white blood cells where white blood cell count goes significantly down such that the person becomes susceptible to everyday diseases that you and I maybe wouldn't be as concerned about. But that leuk has to do with white blood cells. In white blood cells, there's two main components, two main types of white blood cells. Those are neutrophils and lymphocytes. We will go through both of those in a moment. And lymphocytes are made of two types of cells, B and T cells. So we will go through all of those here in a moment. In terms of your immune system, you can break your immune system into two main categories, innate versus acquired. The innate immune system is non-specific and targets any and all foreign pathogens, trying to keep them out of your body to begin with, or trying to destroy them as soon as they get inside of your body. Acquired immunity is specific and targets only one specific pathogen. So we are going through all of this in much more detail right now. First up is the first line of defense. The first line of defense is part of your innate immunity. The goal is keep anything foreign out of your body. Inside your body, you are nice and warm. You are nice and juicy. It's really good conditions for pathogens to grow and reproduce like crazy. So goal number one of your immune system is keep the foreign pathogens out. This is non-specific. So whether I'm walking through your sneeze, my skin is going to keep out any and all of the pathogens regardless of what is being carried in your little sneeze particles. So my skin acts as a barrier to keep anything out. Other examples are like your eyelashes and also your eyelids work to clean your eyeball and keep it clean. Essentially, every hole in your body is an entrance through which pathogens can gain entry into your nice, warm, cozy, delicious body. So every time you blink, your eyeball is being washed with enzymes that will degrade any pathogens. You've got some earwax in your ear canal, which serves as a gooey substance to trap foreign things. Coughing, sneezing, mucus, all do the same thing. Get the pathogen out of the body, so push it physically out. Sweat can do the same. Inside your nose and inside your lungs, you have teeny tiny microscopic hairs, and those hairs work to swish back and forth, trying to push pathogens up and out. So in your innate response for your immune system, the goal is keep anybody foreign out. If that does not work, then the immune system is going to go to the second line of defense. The second line of defense is also non-specific, so it is also innate. Non-specific meaning it will target anybody who doesn't belong. In the second line of defense, we see a specific white blood cell that is called a neutrophil. These neutrophils cruise through your bloodstream 24 hours a day, looking for anything that doesn't belong. If the neutrophil encounters a particle, bacteria, virus, maybe even something bigger that doesn't belong, that neutrophil will use a process called phagocytosis to engulf the foreign pathogen. Shown here in this diagram on the left is that process of phagocytosis. When we talk about a pathogen, a lot of the pathogens are extremely small in relation to our white blood cells. Generally, prokaryotic cells are very small compared to the bigger eukaryotic cell of the white bloodstream. So you can see in this diagram an example of just how much bigger the white blood cell is. So here in that picture, I've got the neutrophil cruising along. It encounters that little red bacteria, realizes he doesn't belong, and phagocytosis means that he engulfs that foreign cell. You can see that here. The bacteria is going into the cell. The white blood cell is then degrading that bacteria, chopping it up into many, many little pieces, and then it is pushing out those little pieces as waste. Since this white blood cell is big, it also goes by the name macro, macro being the opposite of micro, micro small, macro big. So it is a macrophage, phage being something that engulfs. So it is a big cell that engulfs a littler pathogen. The neutrophil doesn't care who this foreign pathogen is, he will engulf it regardless, so he's non-specific. After engulfing that foreign pathogen, the white blood cell degrades and chops it up, and that's inside of an organelle called a lysosome. Lyse means to break. We saw that in hydrolysis when we were breaking polymers back in chapter 3. We saw the word lyse when we said that in osmosis, sometimes so much water rushes into the cell. So lysosome means to break. It's an organelle that will break apart that pathogen. Shown here on the bottom right is an actual picture of that, so you can again see just how big this particular white blood cell is in comparison to like the bacteria cell. Now, how does the neutrophil identify who belongs and who doesn't belong? And that comes from an epitope. An epitope is a signal on the outside of the cell that the cell identifies as either belonging or not belonging. On the outside of a cell is the cell membrane. That cell membrane allows things in and out of the cell. It also has a whole bunch of proteins on it, and those proteins might be receptors, they might be channels like tunnels, they could be pumps to get things in and out of the cell. Some of those proteins can also be used as epitopes, meaning they're little shapes that the body uses to identify as belonging or not belonging. Me, in my body, all of my cells that are me, that make me me, all have the same shape on the outside of them. They all have the same epitope. It's possible that if I walk through your sneeze, I might get some particles or some cells inside my body that now don't have that shape saying, it's me, it's me. So my neutrophils would use that shape to identify that that is a foreign cell and thus it should be killed. One example of the use of epitopes is when it comes to blood typing, donating blood, and receiving blood. You can see in this diagram that there's 8 different blood types. Half of them are positive and half of them are negative. Depending on the blood type you are, your blood cells contain different epitopes that your body can recognize as belonging. So, for example, somebody who is A negative has this little triangle shape on the outside of their cell. If a person who is A negative receives B blood, their body would be like, oh crud, what is this heart? This heart shape does not belong, and their body would fight off and kill all of the heart-shaped cells. Similarly, if a person was A negative and got type AB blood, their body again would be thinking, whoa, what are these hearts? They don't belong, hearts are foreign, kill them all. So they could not receive that AB blood. Somebody who is A negative could get O blood because their body would say, oh, no shape. Okay, I don't see anything wrong here. No triangles, triangles are me. I don't see anything here that doesn't belong, so that's perfectly fine. Somebody who, as another example, is maybe type AB blood, their body knows hearts and knows triangles. So if their body sees a heart, they would say, oh yeah, hearts belong. And if their body sees triangles, they would say, yeah, triangles belong. And if they see no shape, they would say, okay, that's fine too. So somebody who is type AB could get any of those 3 top blood types. We call somebody who is type AB blood a universal recipient because they can receive A, they can receive B, and they can receive O. We call somebody who is type O blood a universal donor because they can give blood to anybody. This is just one example, so you don't need to memorize all the different types of blood, but more think big picture. If a body knows the shape and knows the epitope, it will allow that cell to exist inside. If your body sees a foreign shape, it will try then to kill that particle, kill that cell, kill whatever it is. The difference between positive and negative blood is that all of the people who have positive contain a specific protein, so their body knows and recognizes that protein, and they could receive blood that doesn't have the protein. But the opposite is not true. If I'm a negative blood, my body doesn't know what those green proteins are. So if I get those green proteins, my body would say, oh yeah, these green proteins don't belong. So they would try to kill it. So that's a little bit of an example of epitopes. Those neutrophils are cruising along, trying to engulf anybody foreign who doesn't belong. But those pathogens who are inside of you are going to start replicating like crazy. So just the neutrophils alone will not be a big enough defense to kill the replicating pathogen. So at that point, we have a third line of defense. The third line of defense is called the acquired immune response. At this point, the body has already decided this pathogen is a major problem and we need to get rid of it. So this is going to be an attack against a specific pathogen. There's some vocab here that you will need to know. An antigen stands for an antibody generator. An antigen is a piece of a pathogen that causes a response. So it is an epitope that doesn't belong. Epitopes could be good or bad. They could be on my own cells, or they could be on a pathogen. And so when an epitope is bad, then it becomes called an antigen. An antibody is a protein that my cells are making, and my antibody attaches to an antigen. We'll see this. And the goal of that antibody is to further flag them as foreign. I put the snipers here because my third line of defense is like a snipe attack. They are looking for a very specific target. It's not as though the FBI in this picture would shoot like grandpa crossing the street. They're looking for a specific target. So when I talk about antigens and antibodies, I am talking about targeting a specific pathogen. A little bit more vocab and then the steps of that. The specific immune response is a combination of many different cells working together to target that pathogen. Now, when the neutrophil identifies that there's a pathogen and engulfs him, he kind of acts like a serial killer on some of the shows that I've watched. So if you've ever watched like NCIS or Criminal Minds or Law and Order or any of those kind of like killer shows, in a lot of the episodes, I feel like the serial killer saves a little souvenir from who he kills. And it's totally creepy. Like I remember this episode of Criminal Minds where I used to be slightly addicted to that show, and then I had to cut myself off because I would get too scared. But essentially the premise of the episode was that there was a guy who hunted women and he would save their eyeballs and he would put them inside the stuffed animals that he hunted. So he was a taxidermist and he would go hunting like deer and raccoons and foxes and things like that. But he also hunted women and he saved their eyeballs and would put them into the stuffed deer that he had mounted on his wall. Totally creepy, but he saved a souvenir from everyone that he killed. And the neutrophil is the same way. Creepy. He saves a little bit of who he killed. So the neutrophil, when he engulfs a pathogen, he degrades that pathogen in the lysosome and saves a little piece of it on the outside to show his friends. His friends are the helper T cells. The helper T cells are a little bit clueless. And the helper T cell, can't find the pathogen on his own. He's like, what? I don't know what's going on. But when the neutrophil shows the helper T cell, hey, this is who the foreigner is, this is who we need to target, then the helper T cell can say, all right, let's do this thing. So the neutrophil displays a little piece of the pathogen. It displays an antigen. It displays the epitope from that pathogen and shows the helper T cell. The helper T cell then acts like the game of telephone and goes and tells the B cell, hey, this is who the foreigner is. This is who we need to get rid of. So these are which antibodies we need to make. The B cells are then the cells that make the antibodies. An antibody is a protein. An antibody is not a cell. An antibody is a protein that the B cell makes and then pumps out like crazy. And those antibodies float through the bloodstream and attach like magnets to the foreign pathogen. The foreign pathogen at that point, say this is my bacteria cell, is totally covered in these little antibodies. And at that point, another type of cell called the killer T cell can find the pathogen and kill it. Kind of like the helper T cell, the killer T cell is pretty darn clueless on his own and can't find the pathogen. So the killer T cell needs the presence of those antibodies to know who to kill. All right, so here we go to put all the pieces together. First up, we've got a foreign pathogen. That foreign pathogen in this picture is shaped like a sun. It's a bacteria cell. That bacteria cell is in the body. It's warm, it's cozy, lots of food, gonna start replicating. The neutrophils are cruising along 24 hours a day. They identify, hey, here's a shape. This shape does not belong. So the neutrophil engulfs that pathogen, degrades it, but puts a little piece on the outside to show another type of cell, hey, this is who we are looking for. So that's like step one-ish there. Then along comes the helper T cell. Helper T cell, kind of clueless, can't find the pathogen on his own. But he goes and talks to his little neutrophil friend, and the neutrophil has the piece of the pathogen on the outside of it. So now the helper T cell knows who the pathogen is. The helper T cell is then going to relay that information, and the helper T cell will spread the word to the B cells. The B cells also cannot totally find the pathogen on their own in all cases, so the helper T cells help them out, and the B cell now knows this is who the pathogen is, these are the proteins I need to make. Those proteins are called antibodies. The B cell pumps out a boatload of those antibodies. The antibodies float through the bloodstream and get stuck on to the pathogen. The pathogen is then completely surrounded by antibodies. It becomes almost like disabling to the pathogen because he's surrounded on all sides by these antibodies. So at that point, he can't really replicate or do his normal harm because he's so surrounded by antibodies. When the pathogen is covered in antibodies, then the killer T cell can find the pathogen and actually kill the pathogen, normally by poking holes in its membrane. So all of these cells are working in a coordinated, launched attack against the specific pathogen. To remind you, these cells only really know who the pathogen is because the neutrophil had shown them who the pathogen is. So if I got infected with 2 different pathogens at the same time, my body would have to launch 2 totally separate attacks against each of them. It's not as though once I get sick one time, never in my life would I ever get sick with anything ever again. Obviously, that's not true. So every different pathogen needs its own launched attack. And pathogens are in a fight for survival. They are organisms. They, in terms of natural selection, are changing. They're mutating. They're in a fight for survival. So pathogens can be pretty tricky themselves, and they can change what they look like so that the immune system no longer recognizes them. If you've ever gotten a flu shot, you need to get a flu shot every year. And that's just a guess. The scientists and researchers who made the vaccines are just guessing which strain of the flu do they think is going to be the most common one. But the flu, the influenza virus, is constantly changing its epitopes, constantly changing its sugars, because it's in a fight for survival and wants to get inside and reproduce and spread like mass quantities. So it has to keep changing what it looks like so that the immune system doesn't remember it. During an infection, B and T cells replicate like crazy. One way, kind of a general way to figure out if you're sick or not, is to analyze the amount of white blood cells you have or get a white blood cell count. If you have an elevated white blood cell count, it means that your body is currently fighting something, meaning you have more B and T cells than you normally do. So during an infection, these cells clone themselves so that there's enough of them so they can make enough antibodies, communicate, and kill the pathogen. As I was saying, antibodies attach to only one specific antigen. So every disease, every pathogen has a different shape, and thus your body would need to launch a separate attack onto each different pathogen. You do have memory cells. You have both memory B and memory T cells that can remember the pathogen. There's some diseases that you might get one time and never get again. Like back in my day, it was chicken pox. Now kids get vaccines and you probably had the vaccine. But I had chicken pox as a kid, and they say the chance of getting chicken pox again is extremely low because the chicken pox virus just doesn't change that much, and my body will remember that foreign pathogen, and then it doesn't have to go through all those steps again. My neutrophil can identify pathogen, it's this one, it's chicken pox, and my memory B and T cells would say, oh yeah, we remember this guy. Let's make a boatload of antibodies, kill it real quick. Boom, I never even feel sick. That's what this graph is showing. The first time I get a pathogen, my body is clueless, doesn't really know what to do. Neutrophils are cruising. They start to talk to the helper T cells. Now I'm going to make antibodies. The antibodies will diminish because at some point they've done their job and the pathogen is dying. So I no longer need those antibodies. So the amount of antibodies decreases, but some of those antibodies stay in my bloodstream. They remind my B and T cells about who the foreigner is. Now, if I get that same virus, production of antibodies is way faster and I make way more. So the response time for feeling healthy would be way shorter. Maybe I'd stay feeling healthy the whole time and I wouldn't even know. But this has to be against the same pathogen. So these memory cells are the way that vaccines work. When you get a vaccine, you're getting an antigen, meaning they're injecting just the little sugars, just the epitopes, just the foreign molecules, which we call the antigens. So they're injecting just those pieces into your body. Your body is then undergoing that whole immune response. There's no actual pathogen there because you've only injected a piece of the pathogen, or sometimes it's a dead or weak form of the pathogen. But your body will mount to that immune response, and then you'll have those memory cells that remember how to make the antibodies against that pathogen. So to conclude, again, is that same graph like on the previous slide, just saying that at first, when you first get an infection, you don't have antibodies. Your body makes the antibodies. As soon as the pathogen is gone, your body can destroy and recycle those antibodies because there's no threat anymore. The pathogen has been killed. But upon the second exposure, the response would be a lot faster and a lot bigger, meaning you can make way more antibodies in a shorter period of time. Again, that pathogen would get killed and those antigens, or rather those antibodies, would be degraded and their parts reused. But if I get exposed to a new pathogen, I have to start the whole process all over again. So every antigen, every pathogen starts the whole process all over again.